I am Kumar Challapan. I am the chair of the Department of Tumor Biology here at Moffitt. I have been at Moffitt for just more than 20 years. Before that, I was a faculty member at Columbia University in New York City. So we have been working on different signaling pathways that promote the growth and metastasis as well as drug resistance of tumors. And we have been trying to develop novel drugs to target these processes. So we chose this particular OCT4-YAP1 interaction as a therapeutic target because YAP1 protein, which is a transcriptional co-activator, is involved in the genesis, progression, metastasis, stemness, as well as drug resistance of various tumors. And we have been focusing on non-small cell lung cancer. If I may quickly run through 5 of my slides, I can show you the hypothesis and why we are targeting this particular protein. YAP1, as I mentioned, is a transcriptional co-activator. It is a major oncogenic component of the Hippo signaling pathway. And multiple tumor types show alterations in YAP1 levels. They are elevated. YAP1 levels are elevated in multiple tumor types, starting from glioma all the way down to the bladder cancer and colorectal cancer. The subcellular localization of YAP1 is altered in many cancers as well. And we have been focusing on non-small cell lung cancer because our studies had shown that the YAP1 protein contributes to the growth of the cancer stem cells and also contribute to drug resistance. Several downstream targets of YAP1 protein have been identified, and we identified the SOX2 transcription factor, which is one of the major embryonic stem cell transcription factors that contribute to the stemness of non-small cell lung cancer. So we found that YAP1 is absolutely necessary for that. And our published results show that YAP1 levels are highly overexpressed in multiple types of non-small cell lung cancer. In addition, databases like the TCG as well as the KM plotter show that high levels of YAP1 correlate with the poor survival in non-small cell lung cancer. So because of these reasons, we thought YAP1 would be a good target for drug discovery, and we made some specific efforts to target certain interactions of YAP1. Our studies showed that YAP1 protein, the WW domain of YAP1, binds to the PPxY motif of another transcription factor called OCT4. As I mentioned, YAP1 is a transcriptional co-activator. It does not bind to DNA by itself, and it does not have any enzymatic activity or active motif that can be targeted with small molecules. So the best way to inhibit YAP1 function is to prevent its interaction with its target transcription factors. One of the major transcription factors that is targeted by YAP1 is called TEAD transcription factor, and this region of YAP1 binds to TEAD. And our studies showed that it is a WW domain of YAP1 that binds to a PPxY motif within the transactivation domain of another transcription factor called OCT4. Our studies also showed that this interaction of PPxY motif of OCT4 with YAP1 is absolutely necessary for the induction of the SOX2 protein and for maintenance of the cancer stem cells in non-small cell lung cancer. So because of this, we developed peptides, and the Nick Lawrence lab, our collaborator on this project, developed peptide mimetics or small molecules that mimic this particular peptide. And we focused on the mimetics of the PPxY motif of the OCT4 protein. So based on the structure of the OCT4 protein and the YAP1 protein, the Nick Lawrence lab developed around 400 mimetics of the PPxY motif. And I am showing the structure of some of the active compounds here, SR6086, SR6087. These are slightly older compounds, and SR7052 and SR7053. We find to be highly potent in their ability to disrupt the binding of OCT4 to YAP1. So the question is, what will happen if you disrupt the binding of these two proteins? So I have shown a little bit of data that shows that if you treat the cells with these inhibitors, SR86, SR686, SR6087, and other compounds, the expression of SOX2 goes down as we expected, as we predict. Certain metabolic genes, hexokinase 2, PDK1, PDK2, which are all targets of YAP1 and SOX2, are also downregulated. And here is a Western blot showing that the downregulation of SOX2 protein in the cells after treatment with these compounds. So we have a very nice flow of drug discovery steps which led to these compounds. And we first test the ability of these compounds to disrupt the binding of OCT4-YAP1 in vitro. Then we use cell-based assays to show that the SOX2 expression is suppressed. Then we check cell viability. Then we check the ability of these compounds to suppress the self-renewal of cancer stem cells. So here we are showing an example of one of the compounds, how it potently suppresses the adherence independence growth, which is one of the major metastatic features of cancer stem cells. Here we are showing one compound, SR6087, how it suppresses the adherence independence growth of this of cancer cells. We have done this on multiple cell lines. Here we are showing multiple compounds, SR6086, SR6087, SR6170, how at pretty low concentration, at pretty low doses, how it can prevent the formation of the spheres, which is a marker for self-renewal of the cancer stem cells. So these compounds, as we predict, can suppress SOX2 expression and prevent the self-renewal of these cancer stem cells. In addition, we also check the proliferation of a variety of cancer cell lines, and we find that these compounds are very effective in suppressing cell viability. In addition, we also find that these compounds, SR6 compounds, can very effectively prevent the growth of lung cancer organoids. Lung cancer organoids are probably the best in vitro model to study drug efficacy, and they actively mimic the growth of lung cancers in humans. So here we are finding that at a 5 micromolar dose, SR686, SR687, and other compounds can really prevent the growth of these organoids and essentially kill them. This is a live dead assay, where the cells or the organoids which are green are alive and the ones which are red are dead cells. So these compounds can effectively inhibit the growth of the lung cancer organoids as well. The later compounds, the more recent compounds, these are 7 series of compounds. They are also very highly potent, around 4 micromolar against or 3 micromolar against A549 cells, around 2 to 3 micromolar in the case of IC50 in the case of targeting H460 cells, and around 3 to 5 micromolar IC50 in targeting H1650 lung cancer cells. So the first 2 cells are KRAS mutant cells, whereas H1650 harbors mutant in the EGFR receptor. So the reason why we test these compounds is that one of the major problems that lung cancer patients face is the development of drug resistance. I am not showing the actual data, but we are finding that our compounds can really overcome the resistance to the first generation as well as second generation EGFR inhibitors. And we are finding that these can probably cooperate with osimertinib and promote tumor suppression. So we are expecting them to be very effective against cisplatin resistant as well as other standard chemotherapy agents, as well as tumors that are resistant to osimertinib and other agents. Studies are underway to see whether they can overcome the resistance to the G12C KRAS mutant inhibitors, which have just come to the clinic. So this is another experiment showing how the newer compounds, SR7052, SR7053, at pretty low potency, how they affect the self-renewal of the lung cancer stem cells. So we also checked how these new compounds, do they show any dependency for YAP1 in the cells? Towards this purpose, we generated H1650, the EGFR mutant cell lines, which either express a controlled sgRNA or sgRNA towards YAP1. So these cells have no YAP1 in them. And we are finding that the cells that lack YAP1, they are less sensitive to our drugs, suggesting that our drugs can target lung cancers which express a higher level of YAP1 and perhaps more YAP1-OCT4 interaction. And again, as shown here, we find that these compounds are even more potent than the SR6 compounds in overcoming or arresting the growth of lung cancer organoids. So to quickly summarize, we have found that YAP1 regulates stemness, drug resistance, immune suppression, EMT, and metastasis. The PPxY mimetics we developed can disrupt the binding of OCT4 to YAP1, resulting in suppression of self-renewal of cancer stem cells, inhibition of cell proliferation, overcoming drug resistance. They could also prevent adherence-independent growth of cancer cells and suppress the growth of lung cancer organoids with high potency. So these molecules can be expected to have strong anti-cancer effects against non-small cell lung cancer and perhaps other cancer types. The studies that are ongoing in the lab are examining the efficacy of these compounds in mouse models and trying to see whether they can overcome the growth or whether they can arrest the growth of lung cancer in mouse transplant, xenotransplant models.